Hello, my name is Stacy Madrid. Today's date is November 30th, 2019, and this is week 5. We are doing biochemical testing for microbial identification, the carbohydrate fermentation testing. So with this lab, we are testing carbohydrates, which include glucose, fructose, and mannitol. We're also using the fennel red broth. So as you can see on my table, I have all 6 tubes here. They're all labeled 1 to 3. The first one will be testing glucose, then the second one is fructose, third is mannitol. But we are using 2 different active cultures, and those are the S. cerevisiae and the S. epidermidis. So I do have everything set up. Apologies, my goggles are getting a little foggy. Let's see. Yeah, there's number 1. So I'm going to use these fennel broths. I'm going to put the powder, which contains glucose, fructose, and mannitol. I'm put half of them into each of the tubes, and then I'm going to put my Burnham test tubes, which they already soaked in alcohol. So now I'm just letting them fully dry right now before I could actually place them in the fennel red tubes. I have my alcohol here with my pipettes. So first what I'm going to do, I'm going to go ahead and get my number 1 cerevisiae for glucose, and I have my glucose powder. So I'm going to go ahead and open the lip. I'm going to open the tube. I'm going to put the lip over the flame. Just to go ahead and sterilize it real quick. Then what I'm going to do, I'm going to go ahead and open the glucose and put half of this powder into this tube. Go ahead and do that. Put a little more. There you go. Okay. I'm going to go ahead and put the lip of the tube over the flame again. Sterilizing it. I'm going to go ahead and cover it real quick. And I'm going to go ahead and shake it so it can dissolve the powder. Fully dissolve it. Sorry, my goggles are getting a little too foggy. Okay. Okay, so I believe the powder is dissolved now. I'm going to just let the bubbles come off. Okay, so the glucose is fully into the tube and it's shook. This is the S. cerevisiae. So then what I'm going to do, I'm going to open that again, and I'm going to get one of my Burnham test tubes and actually put it inside of the tube. So what we're going to do is we're going to go ahead and open this again. I'm going to go ahead and lip it so it is completely sterile. Then I'm going to go ahead and get one of the tubes, and it's already fully dry. I'm going to go ahead and put it in here. Okay, I'm going to go ahead and lip it again. I'm going to go ahead and close it. So now I have to make sure that there's no air. And it's facing downward. And I actually do not see any air inside of it. Nope, there is no air inside of it. So then what I'm going to do next, I'm going to get my S. cerevisiae. Oops, I'm going to put it back in here. So I'm going to get my S. cerevisiae, my active culture. I'm going to go ahead and open it. I'm going to go ahead and flip the tube to make sure it's sterile. Let's go ahead and set it. Sorry. Set it so it doesn't fall. I'm going to go ahead and take the alcohol out from one of the pipettes. Go ahead and shake it dry. And I'm going to get the same tube I just put the Burnham tube in. Gonna go ahead and open it. Gonna go ahead and lip it. Put the lip of the tube over the flame to sterilize it. And what I'm gonna do, make sure this is a little more wobbly. So I'm gonna get the active culture. I'm gonna get 4 drops of it. So my tube is full, doesn't have alcohol anymore. I'm gonna go ahead and got some real quick. I'm gonna set this down. And I'm gonna drop 4 drops into this tube. 1, 2, 3, 4. This pipette, I'm going to go ahead and put it in bleach. I'm gonna go ahead and flame the lip of this tube. And I'm going to go ahead and close it. So now this tube is ready for it to be in incubation. Now with this active culture, I'm going to go ahead and lip it as well. Now it's sterile. I'm going to go ahead and close that off so there's no spillage happening in this lab. And then pretty much the same process I did for this number 1 silver stain with the glucose. I'm going to go ahead and do the same thing to the rest of them with their appropriate carbohydrate and the appropriate active culture. Once that's all done, I'll go ahead and get 6 tubes and place it into my incubation for 12 hours and assess the results for carbohydrate fermentation. That's it. Thank you.